So we did skip chapter 7. You don't need to worry about it. It's not part of our standards. It gets just a little more off topic. We just finished up chapter 6 on cells, just like the video. Went over some cell theory stuff, and then right into how cells reproduce, right? Now, your guys' modeling with the DNA is DNA synthesis. It's DNA replication, and we can't see it happening. It happens before mitosis. It happens before prophase. Okay, we'll go over the whole cell cycle. Okay, you guys really want to pay attention to this. This is very basic information that all of you will have an easy grasp on if you pay attention. And then, like I said, not only will you have your test, which can improve your overall test score, percentage or average, but the practicum, this is a big chunk of it, okay? It's using a microscope, so it's going beyond this, but you simply have to identify a cell either in telophase, anaphase, or metaphase on a slide, okay? It is difficult if you haven't been paying attention, or you didn't do a real good job with next class's lab, or you just didn't memorize the differences, okay? Otherwise, though, if you do those 4 simple things, you can get an A on your practicum, okay, which is worth 7% of your overall score. So like I've been telling you guys, you still have opportunities here to get your grade up, okay? Okay, so what's the deal here? For cell division in eukaryotes, we're not talking about bacteria right now. We're talking about eukaryotes. It's the whole cell cycle is pretty complex. And the cell cycle is the whole lifespan of a cell. First, you guys know multicellular organisms usually develop from a single fertilized egg. That's the same in plants. You might not think of plants having eggs, but they do. And cell division also replaces cells that simply wear out. So we're talking about repair, right? So we have reproduction, we have repair, and then the cell cycle is pretty much the same for all cells, all eukaryotic cells that have a nucleus. So as a single cell completes the cell cycle, it becomes 2 new daughter cells. So here's your cell cycle. Ladies, listen. Okay, to here. This is an option. Okay, so the first 4 is when cells, right after they divide, they become the new cell, basically, an offspring. They're living in what's called gap 1, MG, gap. It's just growing, and once it's grown to what it's supposed to be, then it's just doing its job. It's just living. If it needs to divide, then it'll move to the next phase, which is what? Eyes up here, you guys. Synthesis, DNA synthesis, which you guys did with your model today, and that's called the S phase. Then there's gap 2. We already have double the DNA. We're not doing our job anymore. We're getting ready to divide. Okay? So this one is more about a little bit more growth because it has to divide into 2. Also reproducing organelles. Mitochondria is copying, more cytoskeletons being made, more ribosomes, more ER. Everything needs to be made because you're going to split your cell into 2. Then you hit mitosis. And mitosis, listen to me very carefully, is defined as the division of the nuclear envelope or the nucleus, because that's what mitosis is doing. And then you would start over. After mitosis, actually, there'd be cytokinesis, the actual cell, or even during the actual cell splitting in 2. And then there's this gap 0, which is when cells don't ever go to synthesis. Following me, guys? So a lot of them, gap 1, they're not going to divide. The nervous cells are the most famous, right? You can't regrow your brain cells. You can't regrow spinal cells. So they never go to synthesis. They arrest, was what they call it, in the gap 0. Now I want you guys to realize that these 3 things together, is it not going to let me write? I went too fast, I think. Going to let me write? No. Okay. These 3 are included in inner phase. So inner phase, the first part is just the cell doing its work. But during inner phase, if it's going to divide, it does synthesis, then G2, then it moves to mitosis. Okay, so let's hear this.
Eukaryotic cells are always in one of the 3 phases of the cell cycle: interphase, mitosis, or cytokinesis. During interphase, the gap stages G1 and G2 occur where cells grow and synthesize RNA, proteins, and other macromolecules. A cell either prepares for the next S or M phase, or performs the cell's special function in a multicellular organism. The G0 stage is a stopping point within G1. Most cells in adult multicellular organisms are in G0. When a cell in G0 or G1 receives a signal that stimulates it to divide, it passes through the restriction point, R. This point of no return commits the cell to a full round of the cell cycle. After passing the restriction point, a cell enters the S phase, during which the DNA of each chromosome replicates to form a new identical set of chromosomes. Cells pass from the S phase to G2. During G2, the cell prepares for mitosis by synthesizing specific types of RNA and proteins, which will be needed in the upcoming mitosis, or M phase. During G2, the chromosomes spread out and fill up the nucleus. Mitosis is sometimes called nuclear division because the nucleus divides into 2 nuclei with identical sets of chromosomes. After mitosis, the whole cell divides. Division of the whole cell is called cytokinesis. After cytokinesis, each daughter cell enters G1. They may then enter G0 or commit to another round of the cell cycle.
Oops. Okay, so then these are going through what we've already talked about, but I thought it was important for you guys to have the information in a packet. Notice the term here, restriction point. Again, it's right here. So if a cell is going to need to divide, there's some signal was sent, there's been an injury, growth, whatever, then it moves past the restriction point and starts to do synthesis. Okay, we went over that. Okay, a little bit with DNA structure. Okay, adenines, thymines, cytosines, and guanines. What are those? They're nucleic acids. Remember way back, I think what was that, chapter 2 maybe, on the 4 organic molecules. You guys did a foldable. Nucleic acids are built, they build up DNA or RNA. Okay, period. Right? So those are the bases. The little pop beads you guys were using, you were representing those 4 bases that are included in DNA. Okay, you guys notice the pattern? Adenine bonds to what? Thymine. I simply memorize this. You guys, heads up. Come on, wake up. We can do this. I simply memorize this with the word at, A and T. Okay? Some people I know memorize the guanine, the cytosine, because a capital G and a capital C are pretty closely shaped together. But either way, if you know one, you know the other. Oh, real quick here, just a note that guanine, and cytosine are held together with 3 hydrogen bonds. They're stronger bonded than the A's and T's. Either way, those hydrogen bonds need to break easily so that we can do what you guys modeled today. You unzipped your strand. You broke your hydrogen bonds so that replication can occur. Or as you'll see later, so transcription and protein synthesis can take place. But right now we're just going to worry about copying this DNA. You also notice that you could only build the DNA in one direction. If you look up here, you can see how they are definitely flipped from each other. Right? The complementary strand here, you could see that these sugars face a different direction on this side, so they're called anti-parallel. from each other. And that DNA polymerase will only work in one direction. Okay, the replication origin, where the area is going to start being replicated, can happen in many spots along your long double helix. Okay, all our chromosomes, all 46 chromosomes are just the DNA supercoiled, 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 and get as condensed as possible. But when they're working in interphase, even if let's just talk about if they're going to copy themselves, they're relaxed into double helix strands. And because they have to be able to unzip and open up to get a full copy of each other, just like you guys did on your desk. So it happens in many spots. You guys are working with only one area. But if you think we could have been doing that all together, we could have had all our strands all together and be replicating these little bubbles. And eventually, when the whole thing's been replicated, it comes apart and you have your two separate strands. Again, DNA polymerase. What is an ACE? What's that mean? It's an enzyme, right? So the enzyme is helping the reaction occur. It's helping the A's bond to T's. More than that, it's also helping the sugar phosphate bond also. And those are covalent bonds. So you can see this model right here, and again it's showing your replication fork, the opening. You can see how one strand is continuous. This would be your number one strand. You're heading towards the opening. Here, we have to wait for it to open and only go in this one direction. So you'll notice you get these little areas that didn't get made because it had to wait for it to open. And then what will happen is DNA polymerase will come back and fix those little spots. They're called Okazaki spots because that fellow figured it out. And now another thing for you guys to realize, and this is pretty cool, is the DNA, it's not like you ended up with a brand new double strand. Your two strands had half of old DNA in it. You guys follow me on that? You guys with me back to your models? You unzipped your old strand, you built a brand new side of each one. So that's called semi-conservative replication because conserved. Half of the DNA was conserved in the new, which you would call the new double helix. You guys follow me on this? So it's kind of cool. Now, what's even cooler about it is that because of how the DNA is copied, you get very few mistakes because you used an old side to make the new side. You guys following me on that one? Rather than like just go ahead and laying that double strand out on your desk and I tell you guys, okay, build me one just like that. Don't unzip it, don't touch it, just build me one. You're going to be more odds you're going to make mistakes than if you unzip it and you're just basically making the opposite to that strand. You guys following me with this? That's one of the things that's so amazing about DNA is how it copies itself. And it copies itself in such a way that very few mistakes are made when we build it. This shows you about the condensing. So in interphase, if we're going to move past gap 2 and we're going to go ahead and head towards mitosis, you're going to get your DNA is going to start supercoiling, uses some histones. These are proteins to get that strand. You can see over here on the right side to get something very long into something very short and compact by doing the supercoiling into chromosomes. So when it says the DNA condenses, this is what they're talking about. They're just supercoiling into chromosomes. Now, if there was a mistake during the DNA replication, that's just called a mutation. So there's an adenine and the thymine was supposed to put next to it. Well, it screwed up. It jammed another adenine in there, or even harder to do, but a cytosine or guanine. Okay? That's a mutation. They can be actually not cause a problem at all, and we'll talk about that more. Those are called non-harmful or silent, but they could have been harmful. They could now code for a messed up amino acid, or for the wrong amino acid making a messed up protein. Now, with mitosis for multicellular organisms, you're just copying cells. Those cells aren't going to be passed on to any of your offspring. The only time you could pass on any mutations that your body or your cells have made is if the mutation ended up in your sperm or your eggs. And we'll get to meiosis later. So right now, if you have a mutation, you go out in the sun too much maybe, and there's a mutation causes and you grow a little, skin cancer. Hopefully not the harmful one. You're not going to pass that on to your offspring. That was from the environment. Okay, the things that cause DNA to mutate are called mutagens. And then the other crazy thing, here the DNA polymerase is helping the bonds happen, but then it even has a proofreading mechanism. It'll go back across to make sure it assembled it correctly. Again, making less and less chances of mutations. If it finds a mutation, another enzyme comes and cuts the wrong amino acid out and fixes it.
Although many cells don't.
We'll stop right there. But the mutation rate for eukaryotic cells is 1 in a million base pairs. So pretty amazing, and that's why, there's not very many mutations. Okay, so next class, I'll talk to you guys about your flip book. We'll finish a little bit more of this and do our lab. I'm not so sure on the dates. We might need a little more time. We'll see how it's going, but that's our schedule so far. Remember, you guys, we just started our 3rd week. 3 weeks until the semester is over. 1 week is full of final exams. So you basically have 2 weeks in each of your classes to get the grades you want. Okay? And the last week being very important for studying for your finals. Please use your planners right now for all your classes. Have a good day. I'll see you guys Wednesday.
I have some questions on my